What you are watching is GENEXIX-cDT, building a clinical digital twin for an investigational drug development question. The input is deliberately simple. Orforglipron 36 mg oral once daily for type 2 diabetes. No structured form. No manual endpoint selection. No hard-coded disease template. The platform has to understand the compound, the target, the dose, the disease, and the trial logic from one sentence. GENEXIX-cDT converts the prompt into a structured case. It identifies type 2 diabetes mellitus, GLP, one receptor biology, the orforglipron compound, and a compound safety-first pathway with confidence scoring. This is where the system separates assumption, evidence, and simulation. The pipeline then activates the disease and compound modules. It pulls from pharmacology, internal medicine, diabetes guidance, safety logic, PK/PD reasoning, and clinical trial design rules. The key point is this. Every downstream output is traceable back to evidence or explicitly labeled as simulated. The platform maps the therapeutic area automatically. Primary target, GLP-1 receptor. The response logic is now disease-specific. It now thinks in diabetes terms. HbA1c, fasting glucose, weight, beta cell reserve, and responder subgroups. Next, GENEXIX-cDT profiles the compound. Molecular weight, lipophilicity, hydrogen bond donors and acceptors, oral dosing, PK exposure, half-life, and therapeutic index are assembled into the digital twin. This is the compound layer before virtual patient testing begins. The platform then runs a virtual trial across 200 simulated patients. It estimates safety, HbA1c response, severe adverse event risk, cascade risk, and responder distribution. The population separates into high responders, moderate responders, non-responders, dose adjustment candidates, and patients requiring exclusion or closer monitoring. The golden reference layer is the most important part. GENEXIX-cDT shows that 42.5% of clinically significant risk cases would be missed by population average screening. Individual golden reference baselines expose what averages hide: renal decline, hepatic impairment, high exposure, and dose reduction need. The system then explains why the prediction behaves this way. It links drug exposure, target engagement, pathway modulation, disease dynamics, metabolic effects, organ toxicity, and clinical response into a 7-compartment reasoning chain. This is not just an output number. It is a mechanistic explanation. GENEXIX-cDT then generates a development strategy. Then comes the feedback validation layer, the moat. The model compares its predicted diabetes endpoints against published Phase 3 benchmark outcomes. HbA1c reduction, fasting glucose reduction, target HbA1c achievement, near-normal HbA1c achievement, and body weight reduction. The visible mean accuracy is 96.2% across 5 metabolic endpoints. Dose escalation, safety monitoring, dose modification, GI tolerability, hypoglycemia logic, pancreatic enzyme monitoring, cardiovascular parameters, and regulatory questions are generated from the simulated trial profile. The platform is now moving from prediction to development planning. The same twin then moves into operational monitoring. The virtual trial becomes a live simulation environment. 200 virtual patients, 28 days, 35 biomarkers, organ system heat maps, subgroup behavior, deviation alerts, all monitored continuously. A pancreatitis risk signal is triggered. The system immediately begins tracking live deviations: blood pressure, postprandial glucose, cholesterol, HbA1c, lipase, C-peptide, and other biomarkers. The health score starts falling as the risk scenario evolves. GENEXIX-cDT does not look at a single lab value in isolation. It looks for cascade behavior. It tracks whether one biomarker deviation is spreading into a multi-organ risk pattern, and whether high responders, moderate responders, and non-responders behave differently over time. The biomarker trend screen shows the longitudinal view. This is where the platform becomes more than a trial simulator. It becomes a clinical safety monitoring twin that can follow populations, subgroups, and individual risk trajectories across time. At day 28, the simulation completes with active deviations, warnings, and one critical biomarker signal. This is the GENEXIX-cDT concept. Test the drug before exposure. Identify hidden risk before averages miss it. And turn drug development into an explainable virtual trial workflow. GENEXIX-cDT by Xcelenz Global.